Before meiosis, chromosomes replicate during S phase but remain uncondensed. In the nuclear envelope, each replicated chromosome consists of two genetically identical sister chromatids connected at the centromere. Above the nuclear envelope, the centrosome replicates, forming two centrosomes. After interphase, meiosis 1 begins. Unlike mitosis, where there is only one round, meiosis goes through 2 times. In prophase 1, chromosomes begin to condense.
The homologous chromosomes loosely pair along their lengths, precisely aligned gene by gene. In crossing over, the DNA molecules and non-sister chromatids break at corresponding places and then rejoin to the other's DNA. When the bundles of DNA separate, the chromosomes are different to the parental ones, and this process of recombination is how genetic diversity is created. Just as in mitosis, the centrosomes with centriole pairs move apart and the spindle microtubules begin to form. The nuclear envelope begins to break down. In late prophase 1, the homologous pairs move toward the metaphase plate.
In metaphase 1, just like mitosis, the pair of homologous chromosomes are arranged on the metaphase plate, with one chromosome at each pair facing each pole. However, in meiosis, crossing over occurs, unlike in mitosis.
In anaphase 1, the chromosomes move toward the poles, guided by the spindle apparatus. The sister chromatids remain attached at the centromere and move as a single unit toward the same pole, and homologous chromosomes move toward opposite poles.
At the beginning of telophase 1, each half of the cell has a complete haploid set of chromosomes, but each chromosome is still composed of two sister chromatids. The chromosomes decondense and the nuclear envelope and nucleoli reform. Cytokinesis occurs simultaneously with telophase 1, forming two haploid daughter cells.
Prophase 2 is the first phase in meiosis 2. Both meiosis 1 and meiosis 2 have the same phases as mitosis. Once again, the spindle apparatus forms in each individual cell. In late prophase 2, the chromosomes, composed of two chromatids, move toward the metaphase plate. Meiosis 2 is most similar to mitosis. This is because the sister chromosomes are split during anaphase 2, which is similar to what happens in mitosis.
The chromosomes are positioned on the metaphase plates as in mitosis. Because of crossing over in meiosis 1, the two sister chromatids of each chromosome are not genetically identical. In anaphase 2, the centromeres of each chromosome finally separate, and the sister chromatids come apart. Now the sister chromatids of each chromosome move as two individual chromosomes towards opposite poles.
In telophase 2, the chromosomes begin decondensing and cytokinesis occurs. The sister chromatids finally separate, and 4 haploid daughter cells result, containing single chromosomes. Each of the 4 daughter cells is genetically distinct from the other daughter cell and from the parent cell. In mitosis, the daughter cells are genetically identical to the parents and to each other.
The formula to determine the number of different gametes produced by independent assortment is 2 to the n. N is the number of homologous pairs. Since there are 3 homologous pairs, there are 8 ways gametes can be arranged by independent assortment. Non-disjunction is when one or more homologous chromosomes fail to separate during anaphase 1 of meiosis. Some gametes will be produced with one or more chromosomes too many, while other gametes will be produced with one or more chromosomes too few.
For example, a gamete with too few chromosomes produces a dysfunctional embryo. A cell with too many chromosomes can result in a fetus aborted early in pregnancy. One possible consequence of non-disjunction is Down syndrome, which occurs when an individual has 3 instead of 2 copies of chromosome 21.